So I've been looking forward to this talk. I think, I thought about inviting you even last year. And so it's something that I've been really looking forward to. But today we have Jason Knight. He did all of his training in internal medicine, fellowship training at the University of Michigan in Ann Arbor. He is currently the associate director of the lupus program. He's the director of their antiphospholipid syndrome program. He's an associate professor there. He's also a director for the T32 program for scientist training in rheumatology research and the associate chief of research there at the University of Michigan. Today, he's going to talk to us about antiphospholipid syndrome, and I'll let you go ahead and get started.
Oh, thanks a lot. Thanks for the introduction. I'm not sure I've been on the ground at your place since I interviewed for medical school, where at that time they were known for having group interviews, which I don't know if they still do that, but that was a stressor to say the least. A little stressed today, but not as much as I was then. So let's see how it goes. Thanks for letting me talk about my favorite topic, antiphospholipid syndrome, which increasingly, I am kind of putting the title of my grants, not trying to hide it in a lupus grant, and we're getting a little more successful. And so today I will, so here's what I'm going to try to do. So because I can't help myself, I'll tell you some history of APS, give a little bit of clinical teaching. That's not the mainest point of the talk, but I think there'll be some useful bits there. And then I want to get into some of the research that we're doing in APS. So I'll give you a little highlight of our past work and kind of how we got into this field. A few ongoing projects, this clinical trial we have around anti-CD38, and then this wacky story about ginger that I did not set out to study, but kind of fell into. And then I was going to highlight a few things that we'll show at the ACR meeting. A little bit superficial, but maybe will kind of pique your interest to connect with me and or some of my mentees at the meeting in about a month. So let's kind of anchor some definitions. And so as you may know, there have been 3 now international efforts to classify APS, the first in 99 in Sapporo, Japan. This was the meeting held in Sydney in 06, and then criteria just published in 23. So these 06 criteria certainly have some problems, but one advantage is they're kind of simple, like you can show them on a single slide and give people an idea of what you mean. And we'll see in a second with the new criteria, it gets a little more complicated now. As you probably know, the APS field grew out of the lupus field. It was rheumatologists really in the 80s that put this story together, and I'll show you a little bit of that in a second. You know, maybe approaching half of cases of APS will be diagnosed in someone with lupus, but man, there's an awful lot out there that, you may see a little overlap into lupus antibodies, but these are not people with a lupus syndrome. And, we still mostly call these primary APS. I personally have gotten a little bit away from using the term secondary APS. I think it just doesn't put the gravity of APS in the right place for all patients. So in those cases, I talk more about APS and lupus as coexisting diagnoses. So as you probably know, like at a high level, like I think everyone will know this, that clinically we're looking especially for thrombotic events or obstetric morbidity, especially obstetric morbidity, unfortunately, in the later parts of pregnancy, which can be quite devastating. When you show these criteria to patients, like they notice a few things. One is, and it's, I mean, to some extent true for all of our criteria, but they're very reactive. Like we may know you have the antibodies, but we won't classify you as APS until one of these really bad things has happened to you. And it's not only that we won't classify you, but we often won't treat you. We say, well, how do we know for sure you really have the phenotype that is going to get into trouble? We're certainly not going to start you on anticoagulant preemptively, but that doesn't sound great from the patient's perspective. And the other thing, and this has gotten better with the new criteria, is that these are kind of limited in terms of the full spectrum of things that APS can cause. And so, getting in just before the new criteria came out, this is a review I wrote with an obstetrician and a hematologist published early this year. And, one of the things we pointed out was features of APS, and these are some of these, at least, I'm sure you're familiar with, that were not part of those '06 criteria, things like valve disease and alveolar hemorrhage that can have lots and lots of morbidity. And so now we have a new criteria in 23, which I played a relatively small part in, but was happy to be able to see some of the behind the scenes where I learned it is complicated. And so this is now what we are going to be working off of. Hopefully there will not be another 2 decades pass before these are revised and improved. So these are the clinical domains where you should be looking for 3 points. And I just highlighted a few things I wanted to point out. One is from an obstetric perspective, increasingly going to be leaning into things like preeclampsia and placental insufficiency. These are felt to be more specific for APS than these earlier pregnancy losses, which can just have a lot more potential causes. And so you can see that having only an early pregnancy loss would no longer let us classify you as APS. Of course, diagnosis is a different story, and that is to the discretion of the doctor. Here is where we pick up some of those things I mentioned, like pulmonary hemorrhage or APL nephropathy that were not captured in the old criteria. And then some juicy ones down here at the bottom. Valve disease for sure belongs in the criteria, like this comes up too often. And then thrombocytopenia, something we've known since the 80s was associated with APS, finally gets credit in the new criteria. You can kind of notice that range there, not going all the way down to maybe like the levels you would see in someone who has a full-fledged case of ITP, but looking for, so in that case, maybe the person has coexisting ITP and APS, but it's common in APS to see platelets running in that like 50 to 100 range. And so now you would be able to give someone points for that. So in a different area, so here we have the lab criteria. And so here you would be looking for 3 points. And so the biggest change here, I thought potentially there could have been some more innovation here, but really the only change was to deprioritize the IgM antibodies. You can see now that if you have an IgM antibody in isolation, that would actually not be good enough to classify as APS, because we're also going to be looking for 3 points here. Of course, if you have an IgM antibody that causes a lupus anticoagulant phenotype, then that is a potential loophole that would allow you to be captured. And so again, looking for 3 and 3 from each. And if people have questions, we can talk more about these at the end. But so I wanted to show a little history, like these lab criteria. As you can see, they're not really changing from what we have been working with, but where the heck did these come from? How did we get this confluence of tests? And so I think that can only be known if we look into a little bit of history. And so this is a story I would argue that starts with syphilis if we go way back to the 1940s. And this is when it was first learned how to isolate cardiolipin, a phospholipid, from beef heart. This, as you probably know, is very enriched in mitochondrial membranes. And so heart muscle designed to have all that oxidative phosphorylation is a great place to find cardiolipin. And so it turned out this was a really excellent way to screen for syphilis with high sensitivity, and we still use it today. People with syphilis get anti-cardiolipin antibodies. But by the 50s, and you could imagine in this decade, like marriages dissolving because of false positive syphilis testing. But so now by the 50s, there was an official branding. So although sensitivity, great, specificity, not as good. There's lots of infections that can give a false positive syphilis test. But look at this. So way back 75 years ago, it was recognized that lupus was the most common non-infectious cause of false positive syphilis. So many lupus patients have antibodies to cardiolipin 2, our first hint. Meanwhile, there was this kind of parallel story. They weren't really linked up at this time, so you have to kind of watch them independently until the 70s and 80s when they start to come together. But this was the idea of the circulating anticoagulant in lupus. And so, as the name suggests, when the blood was tested, in the test tube, it looked like it was too thin. And initially you find some case reports like this, people with circulating anticoagulant. They had bleeding complications. But eventually, folks got brave enough to write up the true association, which was paradoxical, so anticoagulant, but thrombosis. So these are patients that were not, in most cases, there's some exceptions, but in most cases, not at increased risk of bleeding, but rather at increased risk of clotting. And so it turns out that laboratory clotting tests have lots of phospholipids, and those kind of replace the cells, right? Because now we're working with plasma where all the cells have been removed. And so these phospholipids give a place where the clotting process can begin. And so it turns out that in this artificial system, where you cannot activate those cells and cause problems, but you're just coating up the phospholipids, you artifactually prolong clotting times. And so this was eventually standardized and given the potentially unfortunate name of lupus anticoagulant. Probably every rheumatologist has had to have a convo with a patient that this is not really a our top level test for lupus, even though it's maybe the only one that actually has lupus in the name. And so in the 80s, Alarcon's group in Mexico was doing similar work at the time, but usually Hughes and his group get credit for putting it all together. And certainly they wrote this very important paper with some other people you might know, like Keith Elkin, there in the middle of the paper. And so here was the story. They took 60 patients. So I think these patients all had lupus. So the idea of primary APS was still a few years away, but about half of them had the lupus anticoagulant test and about half did not. And so one association you can see is with false positive syphilis testing with anti-cardiolipin antibodies. And potentially this number higher if we're using modern testing versus relying on this lab artifact. But the compelling association that could not be missed was the association with thrombosis. And then other things. So yeah, nice. This really has stood the test of time, that also seeing the link with pregnancy loss and thrombocytopenia and CNS disease. So pretty cool and impactful work. I guess the most important paper in the field, you could argue. So by the 80s, we had certainly made progress. This lupus anticoagulant business was kind of understood. The link between lupus anticoagulant and antiphospholipid antibodies was there. However, as researchers began to dig in to understand what was going on mechanistically, one thing they found is that if you purified the antibodies, so instead of testing for antibodies like in serum, where you have a whole mix of proteins, including the antibodies, you're now using a pure substance that you actually oftentimes lost the phospholipid binding activity. So that's a concern. And so the question was if there was protein cofactors in serum that were being missed, cofactors that might serve as a bridge between phospholipid and antibody. And so the one that everyone agrees on is another clunkily named factor, also known as apolipoprotein H, classified with those players, but the name we use in medicine is beta-2 glycoprotein 1. And so it was in 1990 that three groups, the Dutch and the Japanese and the Australian groups, all simultaneously published this observation. And that is essentially here that this is a pretty abundant protein in plasma. It's not a rare bird at all. It seems to be designed to find negatively charged surfaces. The classic example of that is negatively charged phospholipids. We put out a small paper earlier this year showing that beta-2 glycoprotein can also bind to negatively charged extracellular DNA, which we think could have some relevance in the context of the neutrophil extracellular traps that we study that I'll introduce here in 5 minutes. But so yeah, these antibodies branded as antiphospholipid, but this is a case where they seem to be engaging with a phospholipid binding protein instead. And so the other cofactor that I think is worth being familiar with is prothrombin, which you've heard of, does not have as clunky of a name. And this is something folks were thinking about back in the 90s. So here's an example of an early paper, but well, in this miniature field, the most important paper, I think, is by Atsumi and colleagues, published in our flagship journal, A&R, in 2000. And so what Atsumi showed is if you made an ELISA that was built to prothrombin alone, you could kind of see an association, like there was something happening there. If you had high antiprothrombin antibodies, there was some tendency that you were going to have a positive lupus anticoagulant, but didn't seem really highly useful in the clinic. But you could contrast that with this so-called anti-PSPT antibody. And so this is now prothrombin coated onto a phosphatidylserine surface. And so, presumably this changes the conformation of prothrombin, reveals some epitope that is now more specific for APS. But you can see here that it now does a lot better. Like if you have high levels of anti-PSPT IgG, you're very likely to have a positive lupus anticoagulant. You can also see, though, that not everyone with lupus anticoagulant is captured by these antibodies, so it's not a perfect surrogate. We know from using purified antibodies that anti-beta 2 antibodies, potentially through a slightly different mechanism, but they can also give a lupus anticoagulant phenotype. So that's the thing to keep in mind about that LA test, that it's really screening for anti-prothrombin and anti-beta 2 antibodies, presumably at high levels. You need a high level to cause that to turn positive, and that's probably why it's, well, that's at least a theory why it often turns out to be the best predictor clinically, the positive lupus anticoagulant. And so, I don't order this anti-PSPT test on everyone, but where I find it useful is where or when you are having trouble fully interpreting the lupus anticoagulant test. That's often in like a hospitalized patient or someone who's already on anticoagulation. Like there's workarounds if someone's on anticoagulation that you can try to interpret it, but it's never as good as having the nice clean test. And of course, if you're a rheumatologist out there in practice, you know how hard all of these cases seemingly are. And so I think it can be extra valuable information in select cases where you're really considering a diagnosis of APS, but you're not getting that objective data you were looking for. So keep it in mind, like here we send it out to the Mayo Clinic where they run the ELISA and get us the data in days, not weeks, typically. So why research APS? Not necessarily for rich and fame, but I think there's important reasons. So stroke and pulmonary embolism, post-thrombotic syndrome, stillbirth, even, even if you weren't convinced that APS, itself justifies your research, these are, major morbidities in the general population. And so I've always hoped that by researching APS, we would learn some things that might apply more broadly. I think that happened a little bit in COVID. I'm not going to talk about that research today, but yeah, some of the things we were studying in APS ended up being relevant in COVID as an example. I mean, if you're in the clinic and you get any number of these patients, I mean, we see this for all of the diseases we see in the rheumatology clinic, but man, they need personalized approaches to treatment. And this is an example of wanting to be proactive and less reactive. We're often defining someone's APS by like the list of bad things that have happened to them rather than defining it in a way that would let us proactively get them on the treatment that prevents said things from ever happening. It's not as rare as you might think. Maybe a little less common than lupus, but more than some of the things we see like scleroderma or ANCA-vasculitis. So it's not the rarest of the rare. And, as such, it's common enough that not just the rheumatologists, but we need to get the word out to the people who are on the front lines that are often going to be the first ones to send the test. Yeah, and so a question that people have had over the years, and it's, population level studies, not so easy to answer. You have some real experts of that kind of work there at your place, but how common are antiphospholipid antibodies in the general population? And so, yeah, I just used that question to show you just for a couple slides, this paper we published earlier this year. This was led by Ray Zhu, who, I really just kind of glommed onto this one. This was his idea and his organization. So he partnered with the Dallas Heart Study, which has been ongoing for several decades. It's a population level study and, as the name suggests, based out of Dallas, oversampled underrepresented populations. And so it's very good from a diversity perspective. And so, yeah, these are patients that have been followed decade by decade, now maybe into the third decade of being followed, and asking if they develop new cardiovascular events. And so, yeah, this is just to give you a sense of the demographics. You can see pretty good in terms of diversity. And so, what Ray did was measure this panel of antiphospholipid antibodies. So you can see that not all of these are part of the classification criteria. There's some like PSPT that I just introduced you, and also the IgA isotypes of beta 2 and cardiolipin antibodies. And so any positive test was found in actually 15% of people. And if you go for a higher titer, such as 40 units, which does show up, so this is the threshold you need to pass to be called moderate, to have moderate positivity, you still find like 1 in 20 folks had this. A hard thing for publishing this paper was people were very, well, the reviewers were very noting that we did not have like an immediate repeat of the testing. So these were kind of single samples collected every 10 years. And so, we just have to say this was a single testing. Maybe some of that 6%, the levels would have disappeared if we could have repeated 3 months later, but just we did not have the samples to do so. We also didn't have the right samples to run lupus anticoagulant testing. So yeah, hopefully not letting the perfect be the enemy of the good situation here. And so this is kind of the unexpected finding, which we need to make some hypotheses now to try to explain this. But so I mean, yeah, they obviously vigilantly tracked all kinds of cardiovascular risk factors, and they've done a lot of higher level testing as well, things like cholesterol efflux. But after collecting, at least for all the traditional risk factors, you can see actually these IgA isotypes still stood out in terms of predicting future atherosclerosis associated cardiovascular events. And so, yeah, composite of stroke, MI, revascularization, and cardiovascular related death. They did not track venous events well, so we can't speak to that at all. So it may be that IgA does not cause the same sticky blood that makes people acutely clot the way that like IgG antibodies do, but is it telling us something about a more smoldering, either reacting to atherosclerosis that's trying to develop, or potentially even a driver? Pretty interesting. I guess when you find something unexpected, maybe, well, at least, yeah, I have a tendency to trust it a little more. We definitely were not biased in this direction. So be interesting to see what this teaches us in the coming years. So yeah, I'll just briefly show you some of our past work, and we'll see. I'll trim off a section at the end if we end up being short on time. So yeah, I got into APS because during my postdoc time, well, / fellowship time, I was working with Mariana Kaplan here at U of M. She's now at the NIH at the intramural program at NIAMS, where she has a leadership position. But we were studying, or she was studying neutrophils in lupus and got me involved in some of that work. And so just to remind you how this goes, and yeah, this one, that neutrophil is the most abundant white blood cell in the human circulation, not actually true in mice, but in humans. They are, they have granular cells are, designed to have pre-synthesized products so that they can respond immediately. And so yeah, neutrophils like platelets are chocked full of granules, making them very excellent first responders against infections. If you want to do things like single cell RNA sequencing, that works against you because they're not as transcriptionally active as other types of leukocytes. And so it's easy to kind of filter out the neutrophils and discard them if you're not, if that's not a focus of your study. So you can say something like this: the human body produces like 200 trillion or 200 billion neutrophils every day. So over our lifetime, probably just like pounds and pounds of neutrophils. And if you write a review article, you have to, yeah, I think this is actually where you're probably contractually obligated. You have to write the term double-edged sword, that, all of that potent stuff makes them good at combating infections, but can also make them problematic if not properly regulated. And so, yeah, a very interesting thing was discovered about neutrophils in 2004. So here's the classic science paper by Brinkman and Zuchlinski and colleagues in Munich. And so, yeah, another paper that's held up pretty nicely. Not everything is totally perfect with where they predicted this might go, but yeah, definitely a paper that has stood the test of time and rightfully has been cited thousands and thousands of times now. And so, yeah, this is their description of nets, which still holds up, that these are extracellular spider web-like structures. And so the backbone of that spider web is actually DNA itself, DNA that had been super tightly packed in that multi-lobulated nucleus that is now spewed out into the world and used as a scaffolding on which other things can assemble. And so, one of those other things is histones, which have some sneaky antimicrobial properties and also pro-inflammatory properties when they're in the extracellular space, and antimicrobial proteins like neutrophil elastase and myeloperoxidase. And so you could imagine that maybe these nets are useful when the organism is too big to be phagocytosed. And there's some evidence that if a neutrophil undergoes phagocytosis, that gives it signals to suppress this net release. But if you're combating a large fungus or protozoan, that may be when these nets are most useful. And so, yeah, right. The early work was by the microbiologist pointing out the good that nets could potentially do, but it didn't take long to start to think of some bad things. And so is our immune system. And a couple of these things are pretty relevant to, to rheumatology in general, but certainly to APS. And so vaso-occlusion is one that we've studied a lot. I think these nets are designed to be released out in the tissues where the infection is being combated. But when they're released intravascularly, you could imagine that this could be a real nidus for blood clotting. And then, yeah, you also know that in rheumatology, we're used to sending autoantibodies against various nuclear stuff. And so now we have a scenario where that nucleus has essentially been spewed out, not in an anti-inflammatory cell death like apoptosis, but rather a very pro-inflammatory one. And so, yeah, interesting to think about these as a source of autoantibody generation, whether that's in RA or lupus or your favorite disease of interest. So this was the first paper we wrote back in 2015. Yeah, first paper, yeah, in this area without my mentor, Shri Yelavarthi, who's still in the lab, led this study. I mean, it was low key, just a 2-person lab at that time. You kind of look for other randos you can like put on the author list, so it's not embarrassing. Yeah, you don't have that many people working for you. But this is an example of some of the data we showed in that first paper. So we can measure something like myeloperoxidase DNA complexes, which still seems about the best we can do in terms of a specific biomarker for nets. So we might call these net remnants. And so these are, it's not every patient, but a subset of patients that rolled into the clinic, often on an anticoagulant. Things seem to be in check, but you could see this smoldering netosis kind of churning behind the scenes. Another thing we found is if you take a APS neutrophil and put it into culture, it is more likely to spontaneously shoot out a net. And so probably that second signal from it adhering to the plate is the key trigger for that. And then one thing that's made our life somewhat easier is that we can take like patient serum from the freezer and we can expose control neutrophils or healthy neutrophils to it, and we can make them more likely to shoot out a net. So there seem to be factors in the serum that are reshaping the neutrophils and their functions. And at least one of those factors seems to be the anti-beta 2 antibodies themselves, that if we purify neutrophils from an APS patient, we can see that there is some beta 2 trying to live there on the surface. And so these, and maybe that's because it's being clustered there by the antiphospholipid antibodies. We see when we get these clusters on the surface, we see activation of innate receptors, at least toll-like receptor 4, and that seems to get the signaling rolling in the neutrophil. So we've also done mouse models over the years as another way to interrogate this. And so we did the same thing that others have done, which is to take antibodies from our patients and transfer them into mice. At least for activating cells like neutrophils, the FC receptor doesn't seem to be very important. It's more about clustering beta-2 on the surface versus FC-dependent effects. And so that may make it more translatable into the other species. But we showed that if you deplete neutrophils, that's a way that you can reduce thrombosis in these mice. We can, from the hospital pharmacy, we buy Pulmozyme, the stuff that's meant to go into the airways of cystic fibrosis patients to dissolve the nets that live there. But instead, we give it intravenously, and that is a good way to reduce thrombosis. So we think it's not that there's a neutrophil floating in the middle of the blood that's shooting at a net, but rather it finds that second signal from maybe a primed endothelium. And so if we disrupt adhesion, that can be a good way to do it. And then boosting intracellular cyclic AMP. And that's something I'm going to circle back to here in a second with this wacky ginger story. Now, this is just, so the point of this slide is to, if you write the review article, you can kind of hype whatever you want. And so NETosis prominently displayed, and it does seem credible that, I mean, really in any form of thrombosis, I mean, probably not in hemostasis, like the good type of blood clotting that stops us from bleeding out. But when you talk about thrombosis, there's just a zillion different scenarios where people have found evidence of nets there. So it's not surprising that they would be doing something in APS. And if they're an end effector, blocking them might be a good way to prevent that final event from happening. But we also know there's other cells that likely have relevance, including two that we're trying to study, endothelial cells and platelets. So I'm learning more and more about the biology of those cells because that's where some of the data are leading us. So just don't want to leave you with the idea that nets are the entire story. And this probably explains, because you've probably seen papers about nets in almost every rheumatologic disease, even some where it's a little hard to figure what they would be doing. Of course, these diseases all look different, so it's not so simple that it's just nets and nothing else, but it's clearly an interplay with other types of cells. Okay, so this will only take a few minutes, but I wanted to, just give you an idea of some of the other types of things we're working on. So if you're a rheumatologist, you certainly know about CD20. Like one of our favorite drugs is rituximab, which targets CD20. But when this has been studied systematically in APS, it is we don't see Rituxan reducing antiphospholipid levels. So this targeting these B cells isn't sufficient to do that. And so what's going on? Well, it turns out cells that don't express CD20 well are actually the ones making the antibodies, whether short-lived plasmablasts or longer-lived plasma cells. But a marker they do have that has been leveraged clinically in the multiple myeloma field is CD38. This is an ectoenzyme involved in NAD metabolism. And actually my boss from your place, John Varga, this is one of the things that he's studying in fibrosing diseases. But here we're thinking more about it as a cell marker. It's expressed at the highest levels on plasma cells. And so it's been leveraged over the years to deplete plasma cells. And so here's one of the drugs that's approved for treating myeloma. So this is a cytotoxic antibody that kills off plasma cells in the context of that plasma cell cancer. And there's, well, it's been a little shouted down, I think, by CAR T cells, which have now taken the lead in terms of the thing people are most interested in. But you can also find reports of daratumumab being used in lupus, where there's several reports, but also this single one in APS. And this is not the perfect study, but just to highlight the concept. So this is a patient, while admittedly getting lots of other convoluted therapies, had daratumumab added to the mix. And indeed, there was a resulting decrease in antiphospholipid levels with an eventual rebound, maybe as the daratumumab effect got out. I mean, this is not the most classic patient, though. The highest level antibody in this patient was PSPT IgM. And so a hint there could be something there, but still needs to be studied systematically. And so that's what we're trying to do. So this trial is being sponsored by the Immune Tolerance Network through NIAID. And so I don't think they've ever done an APS study before. As you know, they're interested in, as you may know, they're interested in not only autoimmunity, but also allergy and transplant. So autoimmunity is just one of the things they look at. But they were kind of looking for a disease where an autoimmune disease where daratumumab could be studied systematically. And we made some case that APS had some advantages for something like that in terms of we have a relatively small list of autoantibodies that need to be tracked. This isn't lupus where there's a zillion different ones that could be at play. And we also don't have the issue of confounding immune suppression. Like often these folks may be on hydroxychloroquine, but if we're talking primary APS, unless things are really going badly, we typically don't have them on other types of immune suppressants. And so it's technically a safety study, but there's lots of endpoints built in that are going to try to tackle the important question of whether, unlike rituximab, this treatment can actually make antiphospholipid antibodies go away. I mean, acknowledging that this is yeah, I don't see a future where we're putting people on chronic daratumumab therapy, but could it be part of an induction regimen that could be followed with a milder drug like belimumab potentially that might be a credible thing that we could see in the clinic? Well, so this is the scheme. You can at least make a case on paper that we don't know for sure what's going to happen when an autoimmune disease is treated with anti-CD38 therapy. There's regulatory subsets that also express CD38. What if those are depleted and things go haywire instead? And so yeah, ITN was very interested in having kind of a dose response or a dose escalation scheme. That's the better way to say it. So 16 mg per kg is the dose that's approved for myeloma. You could also imagine the burden of plasma cells is potentially going to be a lot less in these folks versus someone that has a plasma cell cancer. And so maybe we'll find that a lower dose works just as well. So that's one part of the scheme. And then, yeah, the other part that I said is that well, and we'll see. If the antibodies go away, we're going to have zillions of tubes of blood stored to do interesting mechanistic studies on. If the antibodies don't go away, probably as things go, probably not as much testing will be done, but let's hope they do. Yeah, and we'll also try to do some readouts because we think APS is a disease where the antibodies are not just markers of disease, but actually pathogenic. And if we could cure the antibodies, is that the same as curing APS? So the case that the antibodies are driving things come from stuff like animal studies that I talked about and like the types of in vitro studies we can do where we mix antibodies with healthy cells and show that they change. But we've never in people been able to make those antibodies go away to see if their blood is less sticky, if you will. So I hope this is an interesting opportunity to do something like that as well. And yeah, maybe prove what's truly at the heart of APS. Okay, so here's another kind of quick one. So this is some work we showed at the ACR meeting last year, this story overall, but yeah, but now we have a couple papers out. So this was the first paper that was published back in 21, and this was the idea of Ramadan Ali, who was a postdoc in the lab. Now he's a PhD researcher, now a junior faculty trying to make his own way. And so he's the one that had done some of our work around cyclic AMP and neutrophils. And so at the same time, he's always been interested in natural products. And he found old data that 6-gingerol, which is the most abundant bioactive phytochemical in ginger root, that it has properties like a phosphodiesterase inhibitor. So the one drug in rheumatology I'm aware of that inhibits PDE4 is called apremilast, which, yeah, I guess is approved for psoriasis and psoriatic arthritis, although I'm not sure it's felt by people on the ground to be super effective in those conditions. But where it does seem to work really well is in Behçet's disease. I don't know if officially approved for that or not, but there's certainly been some pretty compelling studies. And so Behçet's, if any disease we see is like a neutrophil, well, you can for sure convince yourself that neutrophils are part of the pathophysiology there. So maybe that's a hint of PDE4 inhibitors for treatment of neutrophil diseases. And so what happens if you give a PDE4 inhibitor is you get higher levels of cyclic AMP in the cell, and this tends to have anti-inflammatory properties. This is a classic second messenger that turns on downstream kinases that tend to have anti-inflammatory effects. So yeah, Ramadan first showed some data like this. He compared 6-gingerol to synthetic phosphodiesterase inhibitors and showed, I mean, in an artificial system, but So here he's treating neutrophils with these compounds and showing a decrease in phosphodiesterase activity and an increase in levels of cyclic AMP. He showed that if he puts this onto neutrophils, he can also, so if he feeds them some ginger, he can also reduce netosis there. Yeah, I'm not going to show you the data from this paper, but it did work well in mouse models as well. But then this is a paper that was just published, just came online a few weeks ago, a follow-up study where instead of focusing on 6-gingerol, now focusing on more of like a proper ginger extract as someone might be able to obtain and consume. And we got this for free from a company in India. They did not have any other input into the study though. And so, they gave us a powdered form that was easy to feed to the mice. And so it was either gavage to the mice or for longer experiments, it was mixed with their chow, and both seemed to work okay. And so here's an example of a thrombosis experiment where, in this panel B, you can see these net remnants in the blood, which go up when the mice get APS IgG, but then go down in the presence of ginger. And then in that panel C, you can see thrombosis data, where thrombosis goes up with APS IgG, down with the presence of ginger. So fine. But what about in human beings? And so we did a small pilot study, which we didn't really have funding for. So this was done with discretionary monies. But here's what we did. We drew people's blood, let them take or asked them to take the ginger supplement for a week, drew their blood again, and then there was a 1-week washout period. And so we measured things like neutrophil cyclic AMP, which clearly went up, like a pretty striking and impressive finding. And I think also notable that this contrasts with PBMCs, where we don't see the same effect. From our mouse data, we have some idea that neutrophils, these phenolic phytochemicals like gingerols are glucuronidated by the body, which seems to reduce their biologic activity. That may be one reason the supplements, you can't always replicate things in humans that you find in the test tube. But neutrophils express a lot of glucuronidases. So are neutrophils like particularly good at eating these things and then reactivating them? Maybe, that's one theory. And then here showing some experiments where he took neutrophils purified from the people and gave them some stimulation, and you could see their responses were more blunted, and then a week later starting to, if not fully, rebound. And so we asked our collaborators in Colorado to see if they could repeat the same trial and get similar results. So this was all done totally independently of us. And honestly, with a slightly different supplement, so that may have been a slight confounder. Also, the clean air and good living there, so who knows what that might be doing to people's neutrophils. But we still got some statistical effects. Not every participant was perfect, but we could still see a difference pre and post ginger consumption. And I think, doing something like this is stressful. Usually you wait for the follow-up paper to see if you can reproduce a big study, to be honest. But I think the reviewers actually liked it, that they were happy to see that we were trying to replicate these findings. So now we're trying to figure out how to get funding to do a larger study in something like APS or rheumatoid arthritis or something else, but so far haven't unlocked those monies, but we'll see. But yeah, so, okay, and we'll see if maybe I have time to get through a couple of these short stories. We'll see how it goes. So yeah, this is a cool and a little bit complicated study being led by Naveen Kumar in the lab, who's a postdoc. But I think I can give you the highlight and maybe make you interested to stop by his talk at the ACR meeting. And so for a few years, we've been studying these enzymes called ectonucleotidases. And so what they do is regulate the extracellular balance of ATP and adenosine. And so, of course, all the things ATP and adenosine do inside the cell. It's lots and lots of things that are fundamental to life. But we also know they have roles outside the cell because our cells have receptors for them on the surface. And a rough rule of thumb that mostly holds up is you can think of ATP as kind of a danger signal, identifies a problem. And in contrast to adenosine, which tends to be anti-inflammatory, one of the things adenosine does is to boost, well, at least the adenosine receptors that are expressed at high levels on neutrophils boost cyclic AMP levels. And that's actually originally how we got into the cyclic AMP studies there. And so, yeah, he really wanted to do something with platelets. And so we decided to do a neutrophil platelet combo project. Like he pointed out that platelets are known for being very good sources of both ATP and ADP. And so you could imagine there might be a role for ectonucleotidases in trying to cool that those pro-inflammatory signals down. And so, yeah, kind of I guess, yeah, always like to do some testing in the patients. Like if we just jump into the in vitro only, like how do we know this has any relevance? So yeah, to really get convinced to do this study, we did, well, not really a pilot study because it was 50 people by the time we were done, but surveyed people that came into our clinic in terms of whether their neutrophils would be very good at hydrolyzing ATP or perhaps somewhat less good. And you can see that as compared to the healthy people, it's not everyone, but there's a subgroup of people that seem to be pretty bad at hydrolyzing ATP. And actually the same was true for their platelets. Again, it's not everyone, but there's a subgroup where their platelets are really bad at hydrolyzing ATP. So you could imagine that, yeah, ATP is going to be pro-inflammatory, pro-thrombotic. You'd like to cool that down and maybe not so good at doing it. And so, yeah, one thing he showed also in the patients is that both for neutrophils and platelets, like this defect in hydrolyzing ATP associated with higher levels of neutrophil platelet aggregates. So that would fit with the idea that these cells are a little revved up if you're not able to hydrolyze ATP effectively. And then here's where I thought, well, now we have something we can tackle in vitro, is because he did kind of a simple experiment where he took healthy neutrophils just minding their own business and gave them a CD39 inhibitor. So no extra ATP added, like any ATP that was there must have been constitutively released by the neutrophils. But you can see that the simple addition of CD39 inhibitor is enough to turn on, well, in this case, neutrophil platelet aggregates, but we can see that with other phenotypes as well. And this especially seems to hinge on the neutrophil side of the equation, that it seems especially that it's blocking neutrophil CD39 that triggers a more aggregate formation and more potential problems. And so, yeah, the whole story is complicated, but in terms of thinking about drug targets, like he has identified some key P2, which are ATP receptors that seem to be required for these effects, a different one on neutrophils and platelets. This may end up being a second story, not in the first paper, but yeah, he's also shown that if he uses adenosine receptor agonists, that's a really good way to cool all of this down. And so, yeah, there's another thing you might think about for therapy in patients. So what the reviewer asked, and rightfully so, and knew it was a hole, and now trying to figure it out, is why exactly these activities go away. Are these enzymes internalized? Are they in intimate association with the antibodies on the cell surface, and that's somehow mucking things up? The fact that we seem to get a similar phenotype in both neutrophils and platelets makes me think maybe there's something kind of fundamental we can discover, but we will see. Okay, so check out Naveen's work. And so here's another one. So this is work by Ray Zhu and Amala Mbati, who's was a rheumatology fellow who just graduated, is now at University of Toledo in a faculty position where we hope to continue down south in Toledo. We hope to continue to collaborate with her around APS. But so here we took kind of whole blood that we had been banking for several years, thinking that we might at some point go back and do RNA sequencing. So this is not, especially if you're interested in neutrophils, I don't think there's any way you could bank the cell samples and go back and do a single cell sequencing after the fact. But doing that kind of thing on fresh blood, I think, will be a future goal. But this is the cruder, old style RNA sequencing. And so it was in about 175 patients. Some of them had positive antibodies, but not clear classifiable APS features. And about a quarter had lupus. So some folks also had lupus. And we just mashed it all together. So here, I mean, as we know, if we compare well, at least like the ANA positive rheumatic diseases to healthy controls, like you get a bunch of interferon signature and everyone gets interferon signature and that dominates everything. But here, we're not using the healthy controls in this part of the study, just comparing these 174 folks against themselves. And, the hypothesis is we would see some type of clustering happening. And there's a few groups on the ends where, yeah, you can see they're very distinct from each other. This group 4 is the one where the net signal based on gene signatures was increased. So these are just different gene modules that were either up or down regulated in the populations. And then kind of for fun was this deconvolution analysis to try to tell us which cells seem to be driving those signatures. So this is not flow cytometry, but this is trying to turn those transcriptomic DNA into turn this transcriptomic DNA or transcriptomic data into kind of a prediction of cell signatures. So again, as you'd expect, we see neutrophils in that group 4. And I guess this is the takeaway, and especially it's especially this point that I think is key, is that, I mean, some of these folks had lupus, some of them had classifiable APS, some not, but really none of those features predicted the clustering that happened. And so, if we had not looked at gene signatures in the blood, we would have thought these folks all looked the same. We had some longitudinal samples that I'm not showing you the data for, but hopefully by the time of ACR, we'll know a little more about whether, the durability of these signatures over time, which is an important point, but nothing quite like this has been done in APS. And so I think it's a first step towards understanding endotypes and better personalized medicine. And I will not go through this full one, but I just wanted to give you the idea. So this has been led by Wei Shi, who was a postdoc and now back in Shanghai, where she has her own lab, and then Wen Ying, who's working in the lab now. And so, just to explain what we're doing, and I won't show you the data, is that we have been biopsying. So we have a very good skin group here that does a lot of high-level profiling, getting into spatial seq now as well, but they're very good at doing single cell sequencing from the skin. And so the hypothesis was that when we see livedo on an APS patient's skin, that tends to predict a more severe phenotype, including certain internal manifestations such as CNS disease. And so the question is, could we look at the blood vessels in the skin, and maybe are they going to teach us anything about what's happening deeper in APS? And so this was the preliminary data that we're trying to turn into a paper, but we now have a grant where we have money now to biopsy 30 different people. And so I think we're going to build instead of going for these people with a super strong phenotype like we did here, we're trying to be more inclusive, all flavors of APS, and we'll see how well. I showed you in the blood, it didn't track so well with what we might have predicted from just knowing the clinical phenotype, but how is the skin going to do? And so we've especially been zeroing in on things like endothelial cells and vascular smooth muscle cells. So I'll skip the data, but we've potentially identified, this is kind of the hypothesis that we think these proteins called CCNs, which are best known for their role in fibrosis, but there's some hints in the literature they also contribute to things like neointima formation, which is the kind of thing you see on APS kidney biopsies. And so, I guess a hypothesis could be that maybe the endothelial cells constantly confronted with blood, and these antibodies are kind of pulling the strings on reprogramming other types of cells that will lead to that type of, not an acute thrombotic lesion, but that more occlusive lesion that we know is, that more chronic occlusive lesion that we know happens in the kidneys because we biopsy it, but it's probably also happening in the heart, in the brain, and other places that we can't biopsy so easily. Well, and then here's some kidneys where we do see like these CCN2 really popping in the kidney lesions of patients. So summary is just reminding you of what the heck I talked about in case you're willing to stay late to ask me a question. This can remind you of what we went over. Acknowledging, I especially highlighted the people in the lab that really led to the research I showed you today, including some key collaborators. So thanks to all of them for doing work while I do paperwork in my office mostly. And then if you're very keen on APS and always want to hear the latest, feel free to sign up for our newsletter, which we try to send out 6 times a year. You just have to give us your name and email. Otherwise, you can look for it on social media and other places. So thank you for inviting me, and thanks for hopefully listening. I can't tell. I could be the only one here right now. Let's see when I escape.
Yeah, that was a great talk. I think it was pretty helpful to get into, you know, antiphospholipid antibody syndrome like that. I'm going to open it up for questions. All right, I think Harris just sent a message that he's got to go, but he can't wait to see you at ACR, so which is great. There's no further questions. I will let you go. Thank you so much for that talk. It was great.
Yeah, find me at ACR or email me if you have any questions.
All
I'd like to
right.
talk about this stuff. Okay, take care. Thanks for inviting me.
Thanks.
Bye.
Christy, did you have a question?
Yeah, I mean, I was gonna just ask, I know this was more like basic science type talk, but I'm curious, I've been feeling for a while that, there's a component of this APS that we're just not treating at all, and it kind of worries me for some of these patients. I'm just curious, how aggressive do you think we should be at this point, especially with like shutting down the inflammatory and immune response? Because at this point, we don't have any studies to guide what to do. But it does worry me. A lot of these young patients, I just see like their cognitive decline. There's just stuff going on that I just feel like we're not targeting with the anticoagulation. And I've tried to get our hematologists and our neurologists to buy into this, and they're sort of very kind of narrow focused in like, well, they don't need any more anticoagulation. But I kind of feel like there's a bigger picture I'm just not treating and we're missing. And I think your studies definitely highlight that. I just wonder at this point, what can I do?
Oh, wow, I feel like this is a planted question because that aligns perfectly with how I think as well. Yeah, I mean, I think there's that unmet need that lives in the microvasculature, and that's why these people get cognitive decline and white matter changes and kidney decline over time. And yeah, I'm submitting a grant. Hopefully, we'll have all the final docs in by the end of the day today. But this is always what I try to highlight in the intro, that anticoagulation is the only thing proven to restrain some of the manifestations of APS, but it's certainly not perfect. I mean, almost everyone in our clinic with bad APS is on hydroxychloroquine, so that's a small one that we use in almost everyone. I think also look for a reason to add a statin in anyone that you can. That's probably keeps the endothelial cells a little happier. But that's where we need, yeah, I mean, because of my interest in neutrophils, like colchicine is one drug. We have a pilot trial concept, like that would be a very safe adjuvant thing that we could add in folks to try to restrain some of the things we're not combating. I mean, when they're having like recurrent alveolar hemorrhage, you put them on something like Cellcept or Rituxan, but I'm not sure it works highly effectively. I think we still don't have it figured out how to best treat these folks. And so it's daunting, but also motivating to try to do better. But I also think it's why, yeah, rheumatologists need to stay in the game because we're used to confusing cases and we're a little more used to living with the uncertainties versus I think sometimes our colleagues in other specialties might want to follow the protocol a little more directly. And we've all been let down by protocols enough that, yeah, we're kind of used to that feeling. So I don't have an answer, but yeah, my thoughts align perfectly with everything you said.
Yeah, I just feel very terrible for these patients because I just see the decline. And
Sometimes it's bad. Yeah, sometimes it's very bad.
Yeah.
And you just watch it continue to. My first APS patient I had when I was a fellow in 2011 just passed away this past year after complications from a kidney transplant. And yeah, I just watched his, I mean, on anticoagulation the whole time, but watching his kidneys decline for a decade. And It's no joke.
Are
Hey,
you?
Jason?
Oh, go ahead. Go ahead.
Oh,
Go ahead,
sorry.
Irene. Yeah, no, go
So
ahead.
sorry, we're over here in clinic, and I'm here with Anisha, and I think we have sort of similar dovetailing sort of questions. So I'll ask mine, and Anisha will ask yours, and it's sort of more like on the complement cascade and how it kind of dovetails with, you know, your work in neutrophils. Like, you know, Jane's work on complement was like so illuminating, right, on like APS and potentially what was happening in terms of like the obstetric complications,
Yeah, paradigm
right,
shifting.
for APS,
Yep.
right? And so, and thinking like, you know, in terms of like an anti-inflammatory potential effect, and, you know, can we maybe look to inhibiting complement and what that effect might be on netosis, and how that might dovetail with like some of the work that you're doing?
No, I think it's a great question. Keith McRae, who's a hematologist at Cleveland Clinic, just reanalyzed some of data we published a few years ago, like the APS neutrophil transcriptome. And yeah, the big takeaway was that C5a receptor signaling was like a huge hit there, and therefore a really attractive potential target. I mean, there was the era when we only had like complement nuclear bombs, like block C5 cleavage, and who knows what the heck else that's going to do. But it seems we're starting to get more refined drugs like C5a receptor blockers, things like this, oral drugs now. So yeah, it's attractive to think about. I think in that subgroup of folks that has microvascular disease, that's where I think the unmet need mostly lies. So companies interested in APS, I think complement is one area that I see. And I mean, just the other area is any drug that reduces antibody levels, because I think pharma is trying to group APS with things like myasthenia gravis, where we think antibodies not just markers of disease, but pathogenic. And so like neonatal FC receptors, I don't know if people are familiar with those drugs, but they kind of force the body to waste extra IgG and have approval in myasthenia, and I think is close in ITP. So in a truly antibody-mediated disease, could that be an approach to treatment? But then complement, I think, yeah, gets downstream of the antibodies, but for sure in a subgroup of people, it's playing a role. So sorry, that was a long answer to a good question.
No, thank you.
I see Irene on the Twitter/X sometimes, so it's good to get a question from her and closer to IRL.
All right, thank you so much, Jason. If anything comes up, we'll give people your email address to
Yeah,
contact
have
you.
them stat email me. Okay, thank you, everyone.
All
Take
right,
care.
thanks.
Bye.